Welcome to Beyond the Threshold, our dPCR podcast. My name is Jonathan, and I'm here with my colleague Jan, and we are both dPCR specialists here at QIAGEN in Hilden. And today we're going to talk about the transition from qPCR to dPCR, or how to transfer assays over from qPCR to dPCR.
Yeah, specifically, this is kind of a follow-up to the webinar we had last year where we talked about the different characteristics of qPCR and dPCR. If you're interested in that, give it a go. You should find the link in the show notes. But this time we're going to focus on people who are coming from qPCR and now might have an assay that they want to switch over. And one of the things that you normally encounter is that the presentation of data, if you just look into the softwares, looks completely different. Maybe you can explain briefly what this all looks like.
Or maybe set the scene first. So you have a qPCR assay, you know that it's running well on your qPCR machine. Now when you want to transfer over to dPCR, you've done the initial setup and you've run your assay. Of note is that usually these assays transfer over fairly well. There's not much adjustment to do just for the basic PCR. You may have to fiddle around with the TM of your assay because the machines behave different. Every machine behaves differently, but it would be the same if you would transfer a qPCR assay to another qPCR machine. So there might be some adjustments, but usually just assume they work as well. You get your data, and then you see a plot and not a curve. The basic difference between qPCR and dPCR is that qPCR is a constant measurement. So after each cycle, you get an image, so then you get a continuous view of what's happening in your PCR and of the fluorescent signal that's been produced. In dPCR, it's different in that you only take a picture once at the end of cycling. So this is an endpoint PCR, essentially, and then at the end of the PCR, you take a picture and look at the relative fluorescence of your partitions. So that's why it's not a curve, it's a plot.
Yeah, and I think one of the main differences for most people transitioning over is if you look at a 1D plot in the dPCR, if you have a lot of target DNA or RNA, whatever you're measuring, you will have a lot of little dots that are higher than the other dots, basically. You'll get two clouds, one is positive, one is negative, and one you have your target. And then the more target you have, the more of those will have a higher fluorescent value versus the qPCR, where generally the more of your target you have, the earlier your curve shows up. And this is normally described by the intersection of a threshold with the curve, which then determines the so-called CQ or CT value, which is the cycle threshold. And this means that if I put in more in qPCR, I'll get the signal earlier, whereas in dPCR, if I put in more, I'll get more dots that are high versus dots that are low on the 1D plot. And that looks completely different. And it is also one of the things where people are sometimes assuming that if you put in more DNA, you will get more signal. In dPCR, you don't. You get the same amount of signal. It's just that you get more dots that have the high signal and less dots that have the low signal. So if you're struggling with distinguishing the two different populations, because maybe your annealing temperature that you mentioned, the TM, isn't quite ideal for the digital PCR application, then you're not going to help it by just pushing in more DNA. You will just eventually end up with no negative partitions, and then you'll just have one cloud, which is not going to help you with the quantification much. So this is one thing that I've encountered a couple of times. And then you really need to work on the PCR conditions, which again, also, if you switch from a normal PCR to qPCR, or even from, let's say, a Rohde gene to another platform from a different manufacturer, you might see differences because even though they all just heat and cool your sample, in the end, that's what a PCR is, heating and cooling cycles, the way that they cool it and heat it might be slightly faster, might be slightly slower. Sometimes the accuracy of these PCR blocks also differs a little bit, and that means that it might be necessary to slightly adjust these, being it to look at the annealing temperature, or maybe also to just elongate the number of cycles that you can run, or that you elongate the extension time or the annealing time for your PCR. There's a lot of variables that you can kind of tamper with. It's the same as for any PCR. If you've ever optimized a PCR in your lab, it's the same for a digital PCR. There's really no difference. It's a PCR reaction after all. But there's a lot of little things that you can twist and turn, little knobs that you can adjust to get the signal better, but just pushing in more DNA, generally speaking, doesn't help. One thing that I've also encountered a lot of times is then when people switch over and they have a good qPCR assay, they might say, well, I've measured this in qPCR and I've gotten 10,000 copies. Now I've measured it in dPCR and it's only 7,000 and I can reproduce this quite well. What could be the issue here, Jonathan?
Well, it's basically the nature of qPCR. You're looking at from cycle to cycle. You're looking if your PCR efficiency is perfect. So 2, you look at the doubling of your template each cycle, and that means the only thing you can measure with qPCR is from one cycle to another, or realistically can do without further data interpretation and algorithms, you can do it from one cycle to another, and that's a doubling of DNA. So that's from 1,000 copies to 2,000 copies, or 5,000 copies to 10,000 copies. And that's the inherent inaccuracy of qPCR. In dPCR, you don't have that problem. The DNA is spread out into the partitions, then amplified, and you just count the DNA templates. That's the nature of dPCR. And so that will allow you to quantify directly and way more accurately than with qPCR.
Yep.
And that's the reason why you thought you have 10,000, but in reality you have 7,000.
Yeah. One of the things that I've also observed a couple of times is that when people work with qPCR to quantify, you always need to compare it with a standard of known quantity. And that known quantity might be not as accurately described as one would hope. I've had some examples. I know that I talked to colleagues here in Hilden when they quantified our AAV kits, and they worked with the virus and they bought a standard that you can commercially acquire, and they always were off by a little bit and they were wondering what's up, and then they asked the manufacturer and they said, oh, well, we print 1,000 copies per microliter on there, but it's actually basically between 700 and 1,300. And then when we adjusted for that, we found out that we were actually quantifying perfectly well. It's just that the number on the vial wasn't accurate. And this is an issue that we're facing at the moment, where a lot of times people use qPCR or even mass measurements to quantify the standards, which are less accurate than the dPCR, meaning that in many cases, the value you measure with your dPCR is the actual number of copies. And then when you use the standard in qPCR and you're assuming a different number that might be printed on the vial, you don't get comparable measurements between your actual samples as well, which is why I've had some customers who said, you know what, if I get a standard, I'm just going to quantify that standard by dPCR, and that way I know how much is in there, and then I can use it for qPCR, and if you then compare the measurements, normally it all perfectly comes out the way that you would expect it, unless your assay has some performance issues or you use a difficult sample type or things along those lines, which can also happen. But normally this means that you get the correct number. So there are some fringe cases, but my experience is a 9 out of 10. If your standard is off, it's probably the standard. It's usually not the dPCR, because it's just robust. It always kind of gives you the same values.
Yeah, I think that's also what I've seen in the field, is the movement toward, or the question becomes more and more like standards. What is a good standard? Where can I get a good standard from? Because nowadays, dPCR being so accurate, there's a real need for more accurate standards alongside, and people are realizing this more and more. So fair point there. Another thing I want to mention is the problem. It's just an inherent how qPCR works is that you have to rely on a good PCR efficiency. If your PCR efficiency is not very well, then your measurements might be off, because it's highly dependent on that. In qPCR, you don't have that problem. Here in dPCR, PCR efficiency only means more relative fluorescent signal. It doesn't have an impact on quantification, and which is, in my opinion, a huge benefit of dPCR versus qPCR. Over qPCR is that you don't really have to care about PCR efficiency, and also therefore you don't have to optimize your assays as much as you would with qPCR, especially when talking about multiplexing or having a reference standard that you have to measure as well, because then you have to make sure that the PCR efficiency for both is comparable in qPCR. In dPCR, not.
Yeah, I think that's one of the things that I've encountered quite often, where people are vastly underestimating how much copies they actually have in their samples, because their samples might be inhibited because you might work with FFPE or stool samples or water or soil or something else, bone, where you have difficulties getting high quality DNA out of there. And sometimes you have PCR inhibitors still floating around in your sample after your prep. And you can mitigate this, of course, by using our QIAGEN kits. You can get relatively good quality DNA out of most sample types. But, sometimes you're still unlucky, or sometimes your sample matrix is just so difficult that you're having a hard time getting all the inhibitors out. And then if you run it in qPCR and you compare it with your standard, which is a synthetic DNA that runs perfectly well, then the same amount of copies will basically show up way earlier in your standard than they would in your sample, because your sample is inhibited. In dPCR, what will happen is you will get less signals. So your cloud of positive partitions will be lower potentially in your sample. But as long as you can still distinguish it from the background, and I think that's what you meant as well, with you don't have to optimize as much, good enough is good enough in dPCR. In qPCR, you need your PCR efficiency to be as close to 100% as possible, because that makes the whole measurement more accurate. Whereas in dPCR, if your efficiency is good enough that you get a halfway decent signal, your accuracy doesn't really get affected. So you can even though your signal might be only 20 RFU above the background compared to 60 RFU above the background for your standard, 20 RFU is still pretty easy to threshold in most cases, and then you can still tell it apart, and you will quantify the correct amounts even though your reaction is inhibited.
So good point here. What do you think? So now I've transferred over, I look at my 1D plot. What do you think a good RFU value is? How would you say, well, I have optimized enough? What's your feeling?
I think we had recommendations once in a handbook, but to be perfectly honest, it really depends on how much you also expose it, because you can change the exposure settings on the QIAcuity at least. So you can put on more light, and the more exposure time you do, the higher your signal will be. That doesn't necessarily mean it's better. The crucial point here is really not so much how much signal do I get, but can I tell positive and negative partitions apart. Now, if I'm only working with a single target in an assay, I only have two populations. That's reasonably straightforward. I just need to be able to put a threshold between those two, and that's really all I need. And generally speaking, the QIAcuity has an auto threshold function that works quite nicely if you have at least, I think, 15, 20 RFU between the two populations. Then it'll just automatically catch it, and you won't even have to manually adjust it. So I would recommend to basically optimize until your auto threshold works. And if the auto threshold starts working, I'm generally do as little work as possible if I can prevent it. We have AI trying to do a whole lot of work for us. The QIAcuity suite also can do a little bit of the lifting for you in this case. And if you can get it to do the thresholding automatically, and you don't have to go into every single well and adjust the threshold yourself, then I would say that is good enough, really. And realistically, I think, like I said, it's about 20, 40 RFU. I think the recommendation is that you should have twice as much signal for your positives as for your negatives, roughly, because it's more of a ratio. Like I said, if I increase the exposure time, both will go up, because if I put more energy in, the fluorophore will give more energy out. So a 2 times ratio would be nice, but like I said, as long as the auto threshold works for most of your samples, I would say that's good enough. One thing I also wanted to mention, because I know a lot of people coming specifically from qPCR are always afraid of increasing the number of cycles that you're running. Because in qPCR, traditionally, there's a lot of applications where if your CT or CQ value is above 35, it becomes a bit fishy. And NTCs at that point, so samples where there's really no template in the sample, like water, start popping up at these values as well in some cases, which is why people don't like to do more cycles in qPCR. And that makes a lot of sense. In dPCR, however, you can just run your NTC, and if that still looks quiet after 55 cycles, then it's fine, even if you have to run 55 cycles. And that can happen if your PCR efficiency is bad, because your sample might be strongly inhibited. I once had a case where basically the assay was very difficult to distinguish, but once we increased cycles to 55, it started working. The reason there was that the amplicon was very long, and it just didn't amplify well.
Yeah. Also, it might well be the case that there are a lot of inhibitors, and the PCR just needs more time to get up there, so you can do more cycle. Another note of the QIAcuity is that when you can do multiple images during a run, you can make if you're uncertain, you can make an image after 30 cycles, and then cycle 20 more, make another image, cycle 20 more, make another image. So you get the full range over you get a broader picture of what's happening during your PCR cycling, if you're unsure of that. Another note, and I mean, we should discuss is the difference in dynamic range. I mean, it's a fair point to say that dPCR has a limited dynamic range compared to qPCR, although and we've done it, and we've said it before in our webinar last year, is we have discussed this is, I think we said something like, it's not that it's smaller, it's just that qPCR dynamic range is so huge that anything looks small compared to this enormously large dynamic range.
Yeah, I think it's also, if you compare it with other techniques that you're measuring, like antibody tests or something, the dynamic range can be significantly less than what you would see in dPCR in some cases. But yeah, it is a point, and in some cases you can compare samples that have vastly different amounts in there in qPCR. One thing to note is there have been a lot of publications as well that show if you have differences that are, let's say, above 5, 6, 8 CQ values in qPCR, you can still see that there is a lot more in there, but the quantification is not as accurate. So you do get more variability and a bigger spread in your repeat measurements when you compare samples that are on the opposite sides of the huge dynamic range in the qPCR. In the dPCR, generally speaking, the dynamic range is shorter, and as long as you stay within kind of the middle ground, you can compare anything that's, you can compare 100 copies versus 100,000 copies, and that's perfectly accurate on both ends of the spectrum. In qPCR, you can compare 100 copies to 100 million copies, but then the fluctuation is much higher. So it's kind of a trade-off. And one thing to note is also that, a million times over expression, when we are looking at gene expression, for example, this is something you normally only get when you artificially stimulate the cells or when you transfect them with something that makes them produce mRNA in ludicrous amounts, because normally physiological changes are usually not that big. If you talk to your physician and talk to them about what's the standard range for anything that they measure in your blood, they'll also not say, oh, this is between 1 and 100 million. The ranges are usually a lot smaller.
Yeah.
Physiological changes are usually within the range that you can catch them with dPCR. Of course, there might be some exceptions, and sometimes when you have a patient that is strongly infected with a virus, for example, they might have huge amount of virus particles in their blood. If they're unlucky, this can happen, so it's not unheard of in nature, but a lot of more normal changes and a lot of things that people are measuring can fit into the dynamic range. You might just have to dilute them. One thing that also I want to mention here is because there is in qPCR, when you do that, when you compare two things that are vastly different in amounts, what you need to do normally is you need to use a primer limited version for the higher one. Now, why is that? In qPCR, I have one reaction bed. So I have all of the PCRs happening at the same time in the same reaction volume. In dPCR, I don't. I spread it out, and then I have one reaction per partition. And if one of my samples is more abundant, then I might have 1 or 2 molecules of that target, and then maybe one of the more rare target inside the same partition. Mm-hmm. But you can imagine that there is way less inhibition if I have 2 molecules of my of target 1 and 1 molecule of target 2 compared to a million molecules of target 1 and 1 molecule of target 2, which means that you get a lot more PCR efficiency drop when multiplexing when you do qPCR compared to dPCR. That's also why it's easier to do in dPCR. But one of the things you can then do is you can use a primer limited version of your higher target, which means that there is less primer and it'll be used up eventually so that your second target can then start creeping up. In dPCR, you just don't need it. And this is something that people ask me, if I reduce the concentration of my higher assay, will I then be able to measure the second one more easily? And the answer is no, you don't. You just have to dilute. And if the difference between the two is too big, you just can't measure them at the same time.
Also, I mean, one point I just wanted to briefly mention that not many people think about is that to even go out of the dynamic range of dPCR for DNA, you need a lot of DNA. And that's something people do not realize. I mean, to overload the system, to overload the dPCR, any of the dPCR systems, you need micrograms of DNA. And that's, I mean, that's not easily obtainable unless you do cell culture or something. It's much easier with RNA experiments. When you're looking at RNA, to overload the system there, you have to be a bit careful. But even if you overload, all you have to do is dilute your sample and just run again.
Yeah, I have to say I have to disagree a little bit with the DNA not overloading. If you work with human DNA, I agree, it's a difficult thing.
See,
But if
okay.
you work with you work with bacteria or if you work with plasmids, it's super easy. I've had this a couple of times when people were using plasmid standards and they're like, how much do I have to dilute this after I've cultured my plasmid? And normally the answer is, I don't know, 1 million times at least.
Yeah, fair point.
Yeah, you get millions and millions, and the thing is that often people measure the mass and then they're like, but it's only 10 nanograms. But yeah, if your construct weighs in the autogram range, that means that there is millions in a nanogram. And if you put millions of copies in, even, at this concentration, to be honest, even a qPCR would be overloaded and you wouldn't be able to measure it properly. But in dPCR also that doesn't work. And that's the thing. So you can overload it also with DNA if you work with small DNA fragments like plasmids or with bacteria or with virus as well. Some viruses are just 5,000 bases, and that's not so long.
Of course, genome. Yes, in this context, genome size matters. So I was thinking about human DNA or animal DNA in general. That tends to be a bit larger.
Yeah, mammalian generally is large enough. Even fish and most of the most of the larger animals also have larger genomes. I know there's the other exception, but across the board, most eukaryotes. It's not so difficult to dilute them down.
So I've transferred my assay over. It's running. I have not overloaded the system. I get a good separation between the positives and the negative. I put my threshold. What's my data output compared to qPCR, where it's a CT value when the PCR reaction passed a certain fluorescence threshold.
Yeah. So the difference really is that in qPCR you get, like you said, the CQ or CT, which is just a value which allows you to compare samples with one another. Where you can say, well, if that CT is one lower, that means I have twice as much in there. So if you measure 2 samples, you can compare these with another. That doesn't tell you how much is in there. If you want to know how much is in there, you need to run a standard. Like we said earlier, this can have its own pitfalls. But generally speaking, the way that most people do it, if they want to know how many copies of their DNA is actually inside, they need to run a standard. And then they need to say, well, my standard runs at the same CT value, more or less. And I know it's 10,000 copies. So then my sample also probably has 10,000 copies. Again, assuming that they both run with the same PCR efficiency, which can be a stretch. In dPCR, you just don't. You just look at the number and it tells you how many copies you have. It's not an arbitrary number. It's just the amount of molecules that are counted by the system, and the analysis is done automatically in the QIAcuity. Granted, if you run a qPCR and you have one of the more newer systems, and newer, I mean, not more than 10 years old, normally they have a function that you can tell them that's my standard curve, and then they'll draw a curve, and then they'll basically link your CQ value to the values on the curve, and you would also get your copies out. But these are the values that you can compare. If you never ran a standard curve, if you don't want to run a standard curve in your qPCR result because you didn't really care how many copies there are, you just wanted to compare samples, stimulated cells versus unstimulated cells, for example, or not knocked down versus knocked down, then you would have to make basically a comparison based on the copies per microliter. So you can say, well, a CQ of 20, like we said, is 1 CQ higher than 19. So my sample at 19 probably has twice as many copies. And then if you take these 2 samples, one at 19, one at 20, this could translate to, for example, 500 and 1,000 copies in dPCR, where the higher one, the one that came up at 19, has 1,000, and then the one that came up at 20 would have round about 500 samples. Now, 500 copies. Now, keep in mind, your qPCR is not going to be less accurate than your dPCR, so don't expect 1,000 and 500. You could expect maybe 1,200 and 300 or something. This would still be what I would say is, to be expected, but it should fit somewhere in the range. You shouldn't see that one of your samples runs at 1,000 and the other one runs at 5. In that case, it would indicate that something is off and either your qPCR doesn't work or your dPCR doesn't work, which it is, kind of depends on the circumstances and how well your assay is defined. Like I said, my experience is it's usually easier to get an assay to behave nicely in dPCR. So I've had a number of cases where in the end it was figured out that the qPCR didn't run as well as it should have, and then they basically reanalyzed their earlier runs. But I've also had the odd case where an assay just doesn't behave in dPCR, and ultimately, in some cases, it was even necessary to redesign the primers and probes. This is something that can rarely happen, but it does happen. It's based on the differences between the techniques. They kind of more or less do the same, but they're not identical. I would say it's like, taking a car to work and taking your bike to work. Both get you from your home to your work, but they work differently, and sometimes there can be discrepancies. Your car might be nicer when it rains, and your bike might be nicer when it's sunny, for example. And this is also the same with PCR assays. Overall they perform kind of similarly, but for some rare cases, some assays might actually work better in qPCR than they did in dPCR because of the way the chemical reaction is going.
Okay. So in dPCR, do I need to use TaqMan probes or probes at all, or can I also use intercalating dyes?
You can use probes if you want to multiplex, because that is not possible with intercalating dyes, because they'll just intercalate into everything. But you can also use intercalating dyes. We have an EvaGreen mix for the QIAcuity. There are other intercalating dye mixes for other instruments. Not all instruments can run with those, but the QIAcuity can, and you could use them if you wanted to. You could also theoretically multiplex, which is something we'll touch on in another episode, I think. But the problem then is if you use intercalating dyes, all of them have the same color, the color of the dye. Normally it's green because it's evergreen or cyber green, meaning if I have multiple primer sets in there, they will all be amplified and they will all be added up, because the QIAcuity can't distinguish what is what, because everything is green. And that means that if I don't care, if I'm measuring two things and I don't care what it is, let's imagine I'm looking for just fungus in my sample. I could run two different fungus assays for two different fungus species, and I could just say, I don't care what is what, I just want to add it up because I want to check for contamination. In that case, I could even multiplex with EvaGreen, but it's kind of fringe cases. Doesn't happen that often.
Would you recommend using TaqMan probes in general? Or would you say, well, if your assay works good enough, you can use EvaGreen and you're not interested in multiplexing?
I would say my experience is that both works. If you just really only need to singleplex, it's fine. But I generally recommend to multiplex, because I've very rarely met anyone who isn't looking at more than one thing at a time. Even if you're only interested in one target, often you have a reference that you need to tie this target to. And then 1 + 1 is already 2, and 2 is easier to do with probes because you can do it in the same well. So as you also mentioned the dynamic range before, and this is something that I also quite often see when people are working with EvaGreen, because often that means that they're also using samples which haven't been as well defined before and in a more research context. Now, how can I spot that I'm outside of the dynamic range of the digital PCR? What does that look like?
Well, unlike in qPCR, where that would just mean that your curve that your CTs get lower and lower and lower. In dPCR, you immediately see that your system is overloaded when you only have one population. And when you only have one population, that means that either you have no negatives, so your system is overloaded, or the PCR hasn't worked at all. In that case, all of them are negative. There is no fluorescence. But it's easy to spot when you don't see two populations of some form or shape. Then you know that your system is overloaded. And overloading a dPCR system does not mean that the RFU values are very high. If you have a good RFU value, so you have a set of bright partitions with lots of fluorescent, that just means that your PCR ran fairly efficient, and you get a good PCR reaction going in your dPCR reaction.
Yeah, that's also one of the reasons why I normally recommend people to run a no template control, usually water. Because that way, if I only see one population in my sample, I can just compare it to the water sample. And if it's the same height, then I know, okay, it's empty. And if it's way higher, then it usually means, I've overloaded and it's actually full. And then you just run a dilution.
Which is actually a good point, because I have seen assays that were negative, but had a so high background fluorescent that they look real. So that the RFU value from the negatives was so high that one could have said, well, yeah, the PCR worked and it's fine. I just don't have empty partitions, but in reality, that was not the case.
Can, for example, happen when you work with probes and you forget to order them with a quencher. Not that that's ever happened to me, though. And with that, I think we can bring it to a close. So thanks so much for listening. Hopefully see or, have you see us at least next time. And see you next time as well, Jonathan.
See you next time, Jan.
QIAGEN.
Sample to insight.